I too would like to thank Sarah for the kind invitation to kick this meeting off. It is truly timely, and it should have happened a long time ago, but I'm glad it's happening now because this is a group of diseases for which there really hasn't been a great deal of attention focused on, and I think your organization is going to shine a bright spotlight onto the diverse conditions that are due to variants of the GABA-A receptor. So congratulations to founding Cure GABA-A variants, and I'm looking forward to great things from the organization. So this is a diverse group. And you bring different levels of understanding. Some of you are internationally recognized experts. Others of you are from the patient advocacy community. And so I'm going to try in the next 20 minutes to kind of set the stage for our discussion this afternoon with a basic review of the physiology and pharmacology and genetics, and maybe some thoughts about where we can go in the future. So maybe there's a problem with the projector, but I think you can probably see that to some extent. The GABA-A receptors are a set of ion channels that sit in the cell membrane, and they're involved in mediating synaptic inhibition. They're the major way in which synapses communicate in an inhibitory fashion. And if you look at the neural circuitry in areas relevant to epilepsy, such as the neocortex or the hippocampus, it's generally organized with neurons that project locally and distally that are excitatory. They typically have a pyramidal shape. You can see them on the slide. And they form the largest group of neurons. But there are also these smaller neurons called GABAergic interneurons that act to inhibit and restrain the activity of the excitatory neurons. So there's a yin-yang balance between the excitation mediated by the principal or pyramidal or relay neurons, as they're sometimes called, and the inhibitory neurons. And there are many different kinds of inhibitory neurons, but they all tend to use GABA-A receptors as their way of communicating with the excitatory neurons and inhibiting their activities. So if we think about this in the context of epilepsy, where we have an altered neural circuit, for example, in the neocortex, we think of the GABAergic inhibitory neurons as providing what's referred to as an inhibitory surround, where they inhibit the excitatory neurons around the epileptic focus, keep it restrained. But when that inhibitory surround breaks down, the epileptic seizure activity can spread and cause an actual behavioral seizure that can affect the patient in a variety of different ways. As you know, there are many different kinds of seizures. As long as the abnormal firing is restrained to a small area, it's not a problem for the patient. We call that interictal activity. But if it spreads, it becomes a problem as a seizure. So the GABAergic inhibition plays an absolutely critical role in restraining activity that is epileptic. And we know how important it is because if you give drugs, for example, bicuculin or picrotoxin, these are often used as rat poisons, actually, because they cause the animals to seize and die. These drugs block GABAergic inhibition. So we've known really since the beginning of neuropharmacology how important the GABAergic inhibition is. Now, this is an illustration of the GABAergic synapse at a zoomed in level. And what you see is that the neurotransmitter GABA is contained in synaptic vesicles in the interneuron nerve terminals. And the GABA is released when an action potential is generated in the inhibitory interneuron, and it diffuses across the synaptic cleft, binds to the GABA-A receptor there, and then allows chloride to flux into the principal neuron. Now, chloride, as is obvious because it's got a negative charge, is a negatively charged anion. And you know that the resting membrane potential of neurons is roughly about minus 70 millivolts or so. And when chloride comes in, it tends to keep the neuron closer to the resting potential and thus inhibits the activity. So this is fundamentally how GABA receptors inhibit excitability in the brain by allowing chloride to flux through the channel. This is an illustration of what inhibitory interneurons really look like. They're very branchy neurons, and they have an axon that synapses onto the principal neuron. And when an action potential, as you can see there, is generated in the inhibitory neuron, it causes what's referred to as an inhibitory postsynaptic potential, IPSP, in the pyramidal neuron. And when those summate, that produces the inhibition. So, I apologize. The slides don't actually look like this, but I think you can get a sense of what's going on there. The GABA-A receptors are what are called heteropentamers. That means they have 5 protein subunits. And there are actually 19 genes that encode these subunits in humans, and they're organized into families, as you can see on the schematic on the right. So you have alpha subunits, you have beta subunits, you have gamma subunits, then you have some other subunits that can substitute for gamma, like delta. But the typical GABA-A receptor consists of 2 alpha and 2 beta subunits. Often the alpha subunits are similar or identical, and the beta subunits may be identical. And then you have this gamma subunit. And the properties of the GABA-A receptors are to some extent altered by the subunit composition. And we'll talk about the pharmacology of the GABA-A receptors. Depending on which subunits you've got, the receptors have different pharmacological properties. What we're showing there on the slide is that GABA molecules can bind at the interfaces between the alpha and the beta subunits. And when GABA binds, that causes the GABA-A receptor to gate open and allow chloride flux to occur. So there are a multitude of potential configurations that you could have with all of these subunits. But there are some generally accepted subunit combinations that are believed to be the major subunit combinations of the brain, and I'm showing those on the right-hand side. I should also point out that there are not only synaptic GABA-A receptors that are involved in mediating inhibitory postsynaptic potentials, as I mentioned, but you can also have GABA-A receptors that float outside of the synapse and are present in the membrane of the target neuron that are not activated by GABA released from the synapse directly, but are activated by GABA that is in the extracellular milieu. And these are called extrasynaptic receptors, and they're believed to play important roles, perhaps in modulating seizure activity. One other thing I should mention is that you're all familiar with the various gene names that are used to designate the specific genes. Each one of those gene names is paired with a subunit protein name, like alpha-1, or beta 2, for example. Each one of these has a specific gene name. And so when a child is diagnosed with a GABA-A receptor variant, it'll be specified with regard not to the subunit name as I'm showing up here, protein name, but rather the gene names. And there's a direct correspondence between those alphabet soup, if you will, and the Greek letters that are used for this protein subunits. So I took this table from one of Catty Kang's very recent publications, where she and her collaborators did a really nice job of summarizing the various epilepsy phenotypes that are associated with variants in these various GABA-A receptor protein subunit genes. And up at the top of the slide there, you can see you've got alpha 1, beta 2, beta 3, gamma 2, and so forth. There's actually some other alpha subunits also that are now linked to various epilepsy phenotypes. But if you go down the left-hand column of the phenotypes, you'll see that there are two broad categories of epilepsies that are believed to be associated with variants of the GABA-A receptor genes. The first ones that were identified were not as really as well characterized as the more recent ones. For many, many years, we've known that it's likely that variation in different GABA-A receptor subunit genes is associated with various types of what are referred to as idiopathic generalized epilepsies. And I'll have to say a little bit about that in a moment. But more recently, it's been discovered, and this is true in many other areas of epilepsy that are related to gene variation. It's been determined that certain more severe epilepsies, which are called developmental and epileptic encephalopathies, are due to variations in the GABA-A receptor genes. So, for example, there are some Dravet-like variants. Of course, Dravet syndrome is most commonly due to mutations in the SCN1A voltage-activated sodium channel gene. But you can also have a Dravet-like epileptic encephalopathy due to variation in the GABA-A receptors, as you can see up there. And this table shows the specific amino acid substitutions that are believed to be responsible. You also have other very severe forms of epilepsy, such as infantile spasms, Ohtahara syndrome, various forms of myoclonic encephalopathies, and so forth. So you really have these two sets of genetic disorders, the idiopathic generalized epilepsies and the DEEs. So there are many clinicians in the room today who can speak more eloquently than I can about the presentation of this type of developmental and epileptic encephalopathy. This is an example that I took from a recent paper where several children with variations in the alpha 2 subunit, the gene name there is GABRA2, were reported. And you can see from the slide that the seizures occurred very early in life, from day 1 to 1 month. There were a variety of different seizure types: clonic seizures, absence seizures, spasm-type seizures, and many of the patients that were described there had status epilepticus. They had many, many seizures each day, and they were highly resistant to anti-seizure medications, which really highlights why we're all here today. We just need better treatments for these severe epilepsies. And we certainly have the option now, or the opportunity, I should say, to really grab at the brass ring, if you will, because we know the genetic basis. And so this is the key to creating precision therapies. And now I think this meeting really is sort of the kickoff of that idea. And I'm looking forward to seeing progress as a result of what we're doing today occur in this area. Up in the upper left, in the upper right-hand corner of the slide, the bars indicate the level of expression of the current that was generated through the GABR receptors with the various variations that were determined in these individuals. And what you generally see is that when you express these mutated channels in Xenopus oocytes and record the chloride currents that go through the channels, you see that there's a reduction compared to wild type. So it's speculated, and I think the data here is not as clean as we would like, but it's speculated that this is a loss of function in many of these situations, at least in this particular study. So there wasn't enough activity of the GABA-A receptors, and that's responsible for the seizure activity in these individuals. So I already mentioned that you can have idiopathic generalized epilepsies, and of course these are the types of epilepsies that are now recognized to be very likely due to genetic variants. They're not associated with any structural brain lesions that can be detected in neuroimaging. And these individuals tend not to have much in the way of symptoms or signs between the seizures. They do, however, have a characteristic EEG. I should mention here that one of the key individuals in this field, who unfortunately is not here today, is Dr. Bob McDonald, who's a good friend of all of us, and who really has been a pioneer for decades on GABA-A receptor physiology and pharmacology. And he and Dr. Kang have been responsible for really defining what the underlying pathophysiological basis for the epilepsies that are associated with these IgE variants. And as you can see, what Dr. Kang and Dr. McDonald discovered is that there can be alterations in the gating of the channel, and there can also be alterations in trafficking of the channels from the intracellular protein manufacturing machinery to the cell membrane. So there are just not enough GABA receptors that are getting to the membrane. And I believe Dr. Kang has also discovered, and I'm sure she'll talk about that later, that the abnormal protein can also damage the neurons as well. Okay, so one other really important thing to mention today is that the GABA-A receptor is one of the most important molecular targets in the brain for CNS active drugs. Many of you prescribe, and perhaps some of you may even take, zolpidem as a sleeping agent. It acts as a positive modulator of the GABA receptor to enhance its activity and thus cause hypnotic effect, a sleep-inducing effect. We also use benzodiazepines like diazepam as anxiolytic agents to treat anxiety. They're highly effective for that purpose. Of course, they have significant problems as well. And we have many anti-seizure medicines that act on the GABA-A receptor We use the benzodiazepines routinely in the treatment of status epilepticus and sometimes in the chronic treatment of epilepsy. Phenobarbital, one of the oldest anti-seizure medicines, acts as a positive modulator of the GABA-A receptor. And I'll mention just quickly ganaxolone because it's the newest FDA-approved anti-seizure medicine, and it too is a modulator of the GABA-A receptor. This just shows you what happens when you take GABA and apply it to a neuron in tissue culture with under voltage clamp conditions where you're looking at the chloride flowing into the neuron, and those downward deflections are the chloride ions fluxing into the neuron through the GABA-A receptors. And if you put on these positive modulators like the anxiolytic diazepam I mentioned or zolpidem, the sleeping medication, you can see that you increase the amount of current. And so this is what we mean by positive modulation. In my lab and many labs, we also look at the activity of the GABA receptors using patch clamp techniques. Dr. McDonald was also a pioneer in this approach as well. What we're showing there is that different modulators can either increase the frequency or the opening time of the channels, and that's basically how they modulate. One of the things that we've done over the years is to try to understand endogenous modulators of the GABA-A receptor. It turns out there are molecules in our body that can modulate the activity of the GABA-A receptors. And the one that we've really focused on the most is derived from the sex steroid progesterone. This is one of the most important steroids for female reproduction. And it goes along with estrogen in the menstrual cycle. Fluctuations occur in the menstrual cycle and also very important during pregnancy. And when we started working on this, it had been recently discovered that progesterone can be converted to a molecule called allopregnanolone, which is also a positive allosteric modulator of the GABA-A receptor. Allopregnanolone can be produced in the ovaries and also locally, actually, in the brain by neurons. When you apply allopregnanolone to a neuron that is being activated with a small amount of GABA, you can see you get a massive increase in the current, and you also markedly increase the duration, the decay time of the inhibitory postsynaptic potential. Now, about 20 years ago, I was sitting in my lab at NIH, and we'd done a lot of work on allopregnanolone. We'd also worked on this molecule called ganaxolone, which is very similar to allopregnanolone, except it has a methyl group in the 3 position on the A ring. And this methyl group allows the molecule to be absorbed orally. It makes it orally bioavailable. Allopregnanolone, if you take that orally, it doesn't get absorbed. We actually founded a company called Marinus Pharmaceuticals to develop ganaxolone as a treatment for human epilepsy. It's been a very long road. I don't have time to tell you about all the twists and turns that we took along the way. But in March of last year, this molecule was the most recently approved drug for the treatment of epilepsy. It goes by the brand name of Ztalmy, and at the moment it's only approved for the treatment of a rare epileptic encephalopathy associated with variation in the CDKL5 gene. But it's being studied for many other conditions, and it actually could potentially be very useful in the treatment of GABA-A receptor variants. In fact, it might actually be more logical to use it there than in CDKL5, for which we actually don't have a mechanism. So I'm going to end with this slide, which is what I use when I lecture to the medical students about anti-seizure medications. You can't really read that, but down at the lower right-hand part of the slide, I list the various different medications that are generally used for the treatment of developmental and epileptic encephalopathies. And again, you can't read it, but what it doesn't show is that there are no real good studies to identify drugs that could be useful for the treatment of the GABA-A receptor variants. And so I'm looking forward to the discussion this afternoon on how we might be able to approach this and develop treatments for these conditions, which sometimes, unfortunately, can be quite devastating. So I'm going to stop there and pass it on to the next person.
All right. Thank you very much.